This is a video about the GCSE Biology topic of classification. It comes up in the second paper of AQA GCSE Biology or Combined Science in the topic about variation and evolution. In this video we're going to describe what's meant by classification and also explain why it's a useful thing to do. We'll describe two systems, Carl Linnaeus' kingdom system including binomial names and also Carl Woeser's domain system, and then we'll explore what evolutionary trees look like and what they tell us. Classification is the process of using similarities and differences to put species into groups based on what each species is most closely related to. And for the purposes of GCSE biology, we're going to say that a species is a group of organisms that can interbreed to produce fertile offspring, what we call the biological species concept. Now, apart from being pretty interesting, classification can be useful from a conservation point of view, but also because it allows us to make predictions about the biochemistry of different organisms, and that can be incredibly important for things like medicine development. A huge number of the drugs that we use in modern medicine have at least their roots in the natural world, even if they're now made synthetically. And one example is the cancer drug Taxol, which is used to treat breast cancer and ovarian cancer. So basically in the late 1950s, early 1960s, the American government were kind of obsessed with curing cancer, and rightly so. And so they got scientists to sample literally thousands of different plants to see if they could find something that would finally stop cancer. And one of the trees that these scientists sampled was the Pacific yew tree, Taxus brevifolia, and they found that it had this really potent anti-cancer compound in its bark that later became the drug Taxol, and it was incredible, and it would just stop the cancer cells from being able to divide. But the problem was that it was only in the bark, and if you strip the bark off a tree then it kills it. And also there was so little of it there that you needed 6 fully grown yew trees in order to make enough Taxol to treat one person, and that's just not sustainable. Now, scientists did try to make Taxol in the lab, and you can kind of do it, but it's a really laborious process, and they just couldn't make enough to make it viable to treat people with it. So they went back to the drawing board, and they started looking at other plants that were closely related to the Pacific yew tree. And they found another plant in the same genus. So this was Taxus baccata, and it makes a very similar compound, although it's not identical, in its leaves. And the great thing there is that you can take a plant's leaves without killing it. So with a small amount of modification in the lab, this molecule could be converted to Taxol, and actually for quite a long time you could literally phone a number and get someone to come and take the leaves of your yew tree away, and they would go and make them into this anti-cancer drug. And that's basically where it comes from. And if we didn't know the classification of which of these trees were most closely related to each other, we'd never have known where to look. Speaking of classification systems, the first one that you need to know about is the one that was developed by Swedish botanist Carl Linnaeus. So a botanist is someone who studies plants. So what he did was he originally split all of the species in the world into one of 7 kingdoms. So animals are a kingdom, and plants are a kingdom, and fungi are a kingdom, and he used common traits to split them up. So for instance, now we would say that all organisms that have a cell wall made of cellulose, a permanent vacuole and chloroplasts belong in the plant kingdom. Even if they're single-celled, even if they live in water, like algae, they're still part of the plant kingdom. And then that plant kingdom can be split into further phyla. So all plants that produce true flowers are placed together in a phylum called angiosperms. And then it kind of carries on from there. So each phylum is split into various classes, each class is split into orders, the orders are split into families, each family contains multiple genera, and each genus contains multiple species. And this is how we classify all of the 8 million or so species that there are in the world. So to look at us, we belong in the animal kingdom. Within the animal kingdom, we're vertebrates because we have a backbone which contains our spine. Within the vertebrates, we're considered mammals because we're warm-blooded and we have hair and we produce milk and we give birth to live young. Then within the mammals, we're primates, which is kind of things that are like monkeys, and then hominidae, which means things that are kind of like men, and then finally Homo and sapiens. You don't need to know exactly which class or order or family humans are in, but you do need to know the 7 layers of the hierarchy in the right order. So kingdom, then phylum, then class, then order, then family, and then genus, then species. You're probably going to want some kind of mnemonic to help you keep these in mind. So I personally use kick fat cat off Farmer Giles' shed. And it's fat with a PH, so that's why he's got his shades on. The last 2 parts of the Linnaean system, so the genus and the species, make up what we call the binomial name for a species. And the idea of this is that it's one system that's used by scientists who speak all different languages and also by different people living in the same country, so that it avoids any confusion about which species we're actually talking about. If you think about the example of a British garden, we have a blackbird. but it probably won't surprise you to know that there are other parts of the world that also speak English, that also have birds that are black, that they call blackbirds, that are not the same as our native British species. So by having different binomial names for them, it allows us to know which one we're talking about. You can kind of think of it as being a little bit like the biology version of the chemistry periodic table, where you have one symbol for each element and it doesn't matter what language you speak, you always know which one you're talking about. When you're writing the binomial name for a species, the genus goes first and is always capitalised, and the species goes second and is not capitalised. And if you're writing it on a computer, then you always write it in italics, and if you're writing it by hand, then you always underline it. The Linnaean system worked perfectly well for hundreds of years, even if we had to do a little bit of shuffling as we started to sequence DNA and started to realise that certain species that looked a lot like each other weren't actually that closely related. But that all started to change in the 1970s. So up until then, scientists had been under the impression that everything in the world was either a eukaryote with a nucleus like animals and plants and fungi, or a prokaryote like bacteria. And there was a group of what they thought were new bacteria that had been found that were called the AQA bacteria, which basically means really old, really ancient bacteria. And these were often extremophiles and were often found in extreme temperatures or really salty environments. But then in the 1970s, evolutionary biologist Carl Woese wrote about the fact that actually the archaea were so different from the bacteria. They were as different from each other as they were from the eukaryotes. They were just two completely different groups and they should never have been put together in the first place. So Woese worried about this for quite a long time, and then eventually in the 1990s he proposed a new system called the three-domain system. So you can see here that the eubacteria, the true bacteria, the original prokaryotes, are in a completely separate domain than the archaea, which we now just call archaea. They're not archaea bacteria because they're not bacteria. And then the eukaryotes. So archaea are still single-celled organisms and they still don't have a nucleus, but in terms of all of the rest of their biochemistry, they're actually very different to the bacteria. You should be able to interpret evolutionary trees to work out which species are most closely related to one another. Here's one example of an evolutionary tree. Along the bottom you can see we've got time, and the branches and the nodes represent how long it's been since two species had a common ancestor. So A, B, C, D and E are what we call extant species, they're still living. Whereas F, which hasn't made it all the way up to the present day, is an extinct species. The shorter the distance we have to go back to find a common node, the more closely related two species are. So A and B are each other's nearest neighbours, and C and D are each other's nearest neighbours. And A and B evolved from a common ancestor more recently than C and D did. And then E is even further back again. These evolutionary trees or evolutionary diagrams are constructed from a whole range of evidence. That could include DNA and protein evidence, so looking at the DNA to see how much it's mutated over time and how it's affected different biochemical proteins, looking at cellular data from microscopy, looking at the whole body morphology, so for instance between two plants we might look at their leaves and see how similar or different they are, and also particularly for extinct species using the fossil record. Thank you very much for watching, and I hope you found that a useful introduction to classification. If you did find it useful, then don't forget to like the video and press the subscribe button below to make sure that you don't miss out on more biology videos coming soon.